Hello, my name is Marilyn McGarrett, and I'm here with Dr. Scheffler. We're going to be discussing the effects of the African swine fever and a biotech solution and how to stop the virus without just quarantining the pigs. So my first question for Dr. Scheffler is, what is the African swine fever?
All right. So African swine fever, it's a viral disease that it affects pigs. It originated in Africa in the early 1900s. And there's been different waves that have gone across the world at different points in history. But right now, it's sweeping through China. It's gotten into Eastern Europe. I've heard reports that it's as close as the Dominican Republic. So it's right on our doorstep. The reason that we're so concerned about it is that it's a fairly deadly disease. So when pigs get it, it might be between 7 to 13 days, and it can be lethal. There are some reports that suggest that in China they may have lost up to 50% of their herd, but again, that's a little bit speculative because a lot of data coming out of China is not the most forthcoming, and so it is a little bit speculation. But certainly in the United States, we're very concerned about it because if that gets on our into our within our borders, then that could be pretty devastating.
And my next question would be, how would we manage the outbreak of the African swine fever using molecular biology? And do you believe a receptor blocker could be used from a different pig species, such as the warhog, the HIV virus, from blocking the human genome to edit out the human embryo from us catching HIV?
So potentially, I know there was some work done, at the Roslin Institute where they were looking at a component of a protein called NF-kappa B, which is involved in the disease response. The idea is that wild hogs and warthogs in Africa, they get the disease, but they don't have it's not you don't get the deadly consequence that you would get in domestic hogs. And so they did some work to see, can we use some CRISPR technology and other techniques to basically change that particular disease gene within domestic hogs to basically impart some resistance to the disease. And they tried it. Unfortunately, it did not work. There was, I think, there was a paper that actually was published here. It would have been 2000, actually just last June, where they tested that particular gene out to see does it in fact impart resistance, and unfortunately, it just didn't work. So we have to identify new targets. So trying to find out other types of pigs that are resistant, try to identify new potential targets that you could potentially modify in the swine herd. One of the challenges that you end up with, though, is in some respects it's a little bit of an arms race. You have a virus that's spreading. Somehow you got to get the defense up before the virus gets here. And so if you develop this gene knockout pig, and even if it's successful, it takes time to distribute that technology to the masses. It's a little bit easier with plants, but with pigs, it's a little bit harder because, maybe I want to develop some boars that have these genetics. Then I got to distribute the semen. It takes 6 months from the day a boar is born until it's reaching sexual maturity to actually produce semen. And then if I get offspring, it's another 6 months before they're on the ground and I can actually propagate it. So in some respects, it's a great technology, but the concern is that it might not be fast enough at the moment. There's some also potential. I mean, if with COVID going around, there's a lot of these mRNA type vaccines, which are based on molecular technology, which is really kind of a fascinating technology because you can take a gene sequence. So, for instance, with COVID, they're using the sequence for the spike protein, and that becomes the target for your immune system, and that's how you can vaccinate against the virus. So far, I haven't heard of that necessarily being used with African swine fever. I just saw a report here just recently that they do have a vaccine that has shown some efficacy in controlling African swine fever. They've actually deployed it in Vietnam in places where there is exposure, and so we're slowly developing tools. But again, it's an arm race. Hopefully, we have the tools before we need them.
There are presumptively mutations that exist in the warthogs and the African bush pigs that don't get swine fever. Can we mimic those genes into our domestic pigs from the warthogs?
Potentially, if we can identify a target that has appropriate efficacy with a lot of these molecular techniques, it's becoming increasingly easy to make these very highly targeted modifications. Right? We don't call them mutations because all we're doing is we're making small edits to the genome to change the DNA into which would allow that animal to better cope with the virus. So certainly, yeah, if we can find those specific targets, we're starting to have the tools to make that change. Just for example, we have in the dairy industry, right? All Holsteins are born with horns. But from a management standpoint, we don't want the horns because, number one, they can cause injury to both people and to, well, other cows. And so what is a long-standing practice is to dehorn, which frankly can be a painful process, right? So if we could go in and just change a single gene, which triggers the growth of horns so that they don't grow them, that's great from a welfare standpoint. There's a lot of advantages to that sort of thing. So that technology is out there. But there's still a little bit of resistance in terms of some of the regulations. I know they've been fighting a battle on if I edit this animal, should that be used for human consumption? Right? There's still a little bit of a fear of some of these genetic techniques, even something as simple as a cow just doesn't have horns. That's the only thing that we're making the change to. So the tools are there. We have to identify the appropriate target. And then the challenge becomes, how do I scale that to a large enough population within the U.S. swine herd to really benefit from having those modifications?
My next question for you is, what are the potential ways that the virus could spread?
So there's multiple avenues that they're currently looking at. A lot of it's through ticks. It could be from direct contact. There's some thought that even if I have a wild pig that was affected with African swine fever, it died, and if that meat wasn't scavenged immediately, that it could actually survive for up to a year. And so there's a lot of different ways that it can spread. And so, it can spread by people. So if one person goes to a farm that has African swine fever and they go to another farm, they might serve as the vector for transmission of the virus because it got on their clothes, it got on their boots, got on their person. And so biosecurity is absolutely critical here. In the 1960s, Spain actually had an outbreak of African swine fever. It took them 30 years to eradicate the virus. And part of the reason is because in Spain they use these Iberian type hogs, which are, they wander through the woods and they eat acorns, and that's what gives it the very distinctive, flavor and texture that you would associate with Iberian pork. But the problem is, in those extensive environments, you don't have any biosecurity, right? They're out in the wild. And so in terms of the U.S., it's really having good biosecurity techniques, preventing cross-contamination between adjacent farms, It's shower in, shower out type facilities where if you go into a facility, you have to shower, change into farm provided clothes to do your work, and then shower on your way out so that you're not taking the virus either way. It could be as simple as asking any feed truck that stops at your place that it has to decontaminate before it arrives and after it leaves. And so there's a lot of we've had we had another virus that was going around the United States called porcine endemic viral diarrhea a few years ago, and they figured out that it was riding on feed trucks. And so once they figured it out, then they had to come up with ways to decontaminate the tires that were rolling through wherever that farm was. Somehow, some way, it picked up the virus. And so now a lot of these facilities, before you drive on the property, you have to start at a kind of a remote location where there's going to be a wash bay, and that's where they're going to decontaminate trucks to try to prevent the spread.
And what could be the possibility of the consequences of how the African swine fever could affect the United States if the outbreak continued growing?
Billions of dollars. I mean, literally billions of dollars. So yeah, if it got into the United States and if it spread like it has the potential to, yeah, it could be a destructive. So pork board. I mean, they're saying a number that's about 8 billion a year, right? So that's a pretty significant number. It'd be great if we could look at what's happening in China, but unfortunately, we just don't have good quality data to say what is actually happening there and what would we expect if it got to our borders. I would like to think that we would have better biosecurity procedures in place, especially since we've already had to deal with things like BVD. But you don't want to take that for granted. So you still want to do everything you can to make sure that A, it never gets here, and B, if it does get here, that we snuff it out as quick as we can.
Thank you so much for your time, Dr. Scheffler. Thank you for answering these questions, and thanks for going over the effects of the management of the outbreak of the African swine fever with molecular biology. But thank you for your time, Dr. Scheffler, and for answering these questions.
African swine fever.